Okay, so good morning, everyone. Welcome to the April ALP2 M4K meeting. Owen, did you have any opening remarks?
Yeah, not really, except for please let us know if this Zoom, this is our, I think, fourth meeting in third different system for seeing how things go. So if you can get any feedback on the system Zoom, if this works well, we're going to open it up to a broader audience for future meetings, etc. So any feedback is always appreciated. But other than that, I think we have a slightly shorter meeting than usual, focused mostly on chemistry, so I'll take it over to you, Jeff.
Okay. All right, great. So I guess I can just open the presentation here. So, you know, I think, again, as Owen said, we have a slightly shorter meeting planned for today. We did have a chemistry meeting earlier in the month where we had some very interesting discussions that we'll touch on a little bit later with the folks at Charles River, Sue and Nicole. And, you know, I think we've got some new and interesting results that have just come out. They're all pretty much hot off the press. You know, we got an RBC report yesterday. We got some PK data yesterday and some cell-based data yesterday as well. So I think it's a good thing to have meetings. the data always seems to come in on the day of the meeting, and so we will do our best to get all of that information out in a suitable format that everyone can open and read and make sense of all of the numbers, because we have M4K numbers, we've got OICR numbers, and we want to be able to get that out to everyone in a suitable format. And but I think we've got some encouraging data. We've got some strange data, and you know, I think it should make for some interesting discussions and for interesting next steps. So again, just for the agenda, I mean, I think it's important for all of us to realize that, you know, we're now about 4 months into the project. I think, you know, again, we're making really great progress. But, you know, Al, I think, sent around an email about a week ago saying that, you know, 4 months is sometimes how long these children survive. So it's very important that we try to speed this work up and go as fast as we can to get a brain penetrant ALP2 inhibitor to treat DIPG. So the agenda is here. I'm going to start just by reviewing some action items from the previous meeting. As well, I'll go over some documentation from the Cancer Therapeutic Innovation Pipeline grant that supports this project partially. And there's some documentation that's due for that. I just want to highlight that for everyone. And then, you know, we'll get into the science with Alex and David Smeal. And then I'll come back with a couple of action items on some of the most promising compounds that we had identified. And I'll show you a little bit of data that we started to generate there. And then we'll finish up with a discussion and any other business. So again, I think we should be done before noon, but you know, I think that depends on the discussion. All right. So the action items from the previous meeting held on March 7th. We talked about following up with RBC. As you all know, RBC has graciously been testing our compounds, and you know, in exchange, we were going to provide them with additional potential assays and targets for assays. And this is something that Alex was going to follow up on at some point. We also wanted to set up a chemistry team meeting between Charles River and OICR. That was done. The meeting was held on March 28th. I think, you know, and Sue, feel free to comment there if you like, but you know, I think we had some really good discussions around the current series of molecules and some of the SAR that we've generated to date. We looked carefully at a lot of different ideas, and generally we came to the consensus that, you know, we agreed to get more SAR information, in particular with this additional set of compounds that we just got data for. And before prioritizing amongst some of the ideas that are already out there. So I think we all agree that the plans that we've made so far all look good, and that we're making good progress. So hopefully we can look more carefully at some of these ideas as more data comes in. But we're really still in that early data gathering stage of the SAR, where we need to learn what's tolerated in the pocket, and what gives us potency and selectivity, certainly against L5. So, we're still in the early days, but I think we're making really good progress, and you'll see a lot of that in David Smil's presentation. And as well, we talked about making sure that there's no duplication of effort, and as of right now, we don't have any issues with that, and we will continue to make sure that we don't have issues with duplication of effort across sites and in similar chemical series. Charles River has set up a portal that allows us to share data, and so we talked about providing some of the names and emails for access to this portal. We've done some of that. I think David and Owen and Arish and I all have access so far. If anyone else wants access to this, please just send me an email, let me know, and we can set that up very easily. And then we also agreed to start scheduling these meetings on our calendars, so that we all know when they're going to occur. And so Angela's been a really big help there, and we've set meetings all the way up until September. So we have dates. I hope everyone is able to make those dates, but certainly we understand that during the summer months there will be some people that are away and some people that just won't be able to make it for some of the meetings. So please do what you can to be present, but we certainly understand if you can't be. And then one other action item was I presented some data on an efficacy study, an unpublished efficacy study at the last meeting, and I made a mistake in terms of pointing out that the cell line that was used in that study, and indeed when we look back at the data and we talked to the people that have generated the data, that efficacy study was done on the cell line that showed the most potency in an in vitro setting. And so I apologize for that error on my part in my last presentation. So hopefully, by identifying more potent analogs, we can get back into an efficacy study and show good results. Now, this efficacy study and the DIPG lines, these are very difficult and aggressive models, and so the fact that we saw anything with an ALK2 inhibitor, I think, is very, very positive news for the project. So, stay tuned, and I think we've got some molecules that are moving through our pipeline to be able to again move into an efficacy study to see how we would do against the lead compounds. All right. So just to remind everyone that this is, and I show this slide kind of at the beginning of every meeting, the deliverables and milestones. And I'll revisit this a little bit in the coming slides when I talk about the documentation that's due for the project, and we may need to have some discussions around this, Owen, in terms of how we're going to handle this. But basically, from months 1 to 18, we're looking at SAR and profiling analogs in terms of the in vitro ADME and the selectivity, and looking at cellular efficacy and mechanism in terms of the phospho-SMAD, looking at inhibition of DIPG cell lines and patient-derived cell lines, looking potentially at some combination studies. And then, basically around the 1 year mark is when we're starting to think, is when it was initially thought that we would start being able to look at some in vivo experiments, especially with respect to mouse oral dosing. And in fact, I'll show you today that we've already dosed a couple of compounds and we have some results to share already. So, I think we're moving along pretty well. Backup series SAR, this is something that we'll need to address at some point, but you know, I think for right now, the entire emphasis is on the lead series of molecules to try to get a proof of concept molecule as fast as possible. So we'll retouch on this again in a couple of slides. So as I mentioned, this project is partially funded by a grant from OICR, the Ontario Institute for Cancer Research. That's the Cancer Therapeutics Innovation Pipeline initiative. And part of that initiative is twice a year documentation that we have to provide. So we have essentially 6 pages that we'll need to write. 1 page summarizing the project goals and hypothesis as was originally stated in the original application. And again, I think this is something that we can pull together quite easily, this 1 page. And I will take care of that. We also need a progress update that's 5 pages long, describing any developments. So for this, you know, I've already talked to Alex about, you know, and he's actually already provided a summary of the biology work and the X-ray work that's been done to date in the project. David will help me write up the SAR portion of the project, and then I'll write as well kind of a summary in terms of status and where we need to go. The big thing, though, and Owen, this is something that we'll need to discuss, and maybe potentially some other people on the team, is that we originally, in the original application, put milestones only 1 year out, and OICR is now asking for milestones every half year, essentially, for earlier quarters. So I think this should be relatively easy, and in fact, we're well, you know, I think we're progressing well against all of these, but it's something that we'll have to discuss.
Yeah, I mean, I think my takeaway on this is there are a couple of things. The project so far is off to quite a good start, to be blunt, I believe. I mean, there's way more green on these charts than I'm used to at anything that's at the 3-month, 4-month stage. So that's really good to see. So people should be happy about it. I think the big point that we're going to have to do, talk to them and make sure everyone's on side, is we want to move out the in vivo work. I think looks like we're going to be pretty close to that. So I think, we already have done some. So I view this as, so far we have a really good story for considering the short period of time. I see this as the bureaucracy we have to go through and the box we have to tick off. But hopefully it won't distract everyone from the good work that we're doing and we just charge on board.
No, exactly.
I mean, we've all done these things in our lives where there's a dramatic change to something that's not the case here. So we'll move forward through this problem. Oh yeah, no, and I'm not concerned. I think if we make faster progress than what was originally anticipated, that's not going to be an issue. So anyway, I just, you know, this is a little bit of an aside, but I did want everyone to realize that this documentation is happening. So if I do reach out for some information, you know, don't be surprised, please. Okay. And with that, I think that ends my initial section, and we can maybe move on to Alex.
Okay, Jeff. Thanks.
What's that? Alex, if you have your presentation open, you should be able to go to the bottom of the top of the screen and share your screen.
I'm on a different computer. Can you share it from your side, Jeff?
Oh, I sure can. Yeah. No, I have it open just in case, but yeah.
Thank you. And you can jump to
this one?
Yep.
Oh, there you go. That's the old one?
No, that's good. That's good. I've just not updated the front page date. Okay.
Yeah, that's the. So I've given you control. You should be able to change the slides, I believe.
Perfect. So I'll just explain. We've got a few people here in Oxford. We've got Paul Brennan with us, who helped write the grant. And then I've got a couple of postdocs and a student. So we've got Roz, John Fu, and Liz here. And I'm going to present all their work today quite quickly. It's not working.
Great.
It did work. Okay. So just a brief word on the crystallography. This is what Roz is doing. Currently, our synchrotron is closed. We've got a trip on the 13th of April, so we're hoping to take some crystals in a week or so, and then we'll collect data, hopefully. This is just an example on the bottom with the arrow just showing some of the crystals we can grow just in the last couple of weeks. But this is just a reference compound. We go to the next slide. Some of the M4K compounds we've been looking at, growing initial crystals. The crystals at the top, the best diffraction there was 3.5 angstrom, so that's still a work in progress. And I think we'll constantly review the IC50 data to kind of select what compounds are really best looking forward.
Right, that was something that I think I noticed as well, that we should review this on a regular basis as new results come in. Yeah, right?
The IC50s are coming in faster than the crystallography, so that will probably change. So again, we're getting some initial bits and pieces. So on the top left, for example, you can see little needles. So if we can optimize the size, we could get data there. Oh, the text on the top's got a bit. So this was an interesting compound with this methyl on the pyridine here. So fingers crossed we can maybe optimize some of these crystals to get some data. And again, we've got little needles with this compound on the top left, which could be followed up. So a work in progress. Just on terms of resource, on Monday next week, we have another postdoc join us, Ellie Williams. So Ellie's been a crystallographer with me for several years on the FOP disease project. And so she can also give us some extra help and service. So we've got options to try and get crystals in multiple ways. So we can do crystal structures of the kinase domain, as on the right, but we can also do complexes where we have two domains, a GS domain as well as the kinase domain, and then combine that with the protein FKB12, and that gives a kind of quite stable protein complex. And so we've got 2 bites at the cherry to get crystals.
And Alex, have you, in analogs that you've looked at before in these 2 different modalities, have you seen essentially good correlation between the 2?
Essentially,
in terms of the binding mode and the kinase domain?
Yeah, in the ATP pocket, we really don't see much difference in terms of any conformational change. They seem quite stable. It's just obviously having the FKBP12 on the top, they just give different packing crystal contacts. So you might get a different crystal. But yeah, we've had very good success with the kinase domain alone, which is why we pretty much use that as our workhorse.
Okay, great.
I'll move on to the cellular data. So this is just a reference of sort of the effects of, on the left is our initial lead molecule that was put in forward for this grant and was used in the in vivo model. So here we have the methylpyridine. And we'd seen in the nanobret, and we just touched on this last time, that while there's good selectivity between ALK2 and ALK5, the drop-off in cellular activity goes from 176 nanomolar on the left to 24 nanomolar if we take away the methyl on the pyridine here. There is a cost to that. So you might want to remember these 2 numbers just for reference for the next compounds coming up. Okay, so this is just substituting on the solvent channel side. So generally I've put the best compounds on the top, going to the worst on the bottom. Again here, substitution of our last group. We have a modest change in affinity, but not too bad. And then reducing the length of the chain on the solvent channel does have a hit on potency. And then some substitutions have obviously not worked. Okay. And then again, in the core SAR, as you probably will find from the reaction biology data too, some of the compounds that we've synthesized recently, or you guys have done, look actually quite promising in terms of the nanobrett in cells, so down to 28 nanomolar. The methyl on the pyridine at this unusual position here, again, seems well tolerated, and having this extra nitrogen has also been tolerated. Okay, so I think those data probably agree quite well with reaction biology data. Some of the last compounds that David and you guys sent over the last week or so have also looked quite promising in the nanobrett assay. So 33 nanomolar again for the top compound, 74 for this second compound. So modifications to the solvent channel here are obviously proving to be tolerated. Of course, our objective has been to also address the trimethoxyphenyl group. Some of the substitutions here have had a drop-off in affinity, so the top ones around 244 nanomolar, we're still seeing some good selectivity, but I think we've still got work to do in terms of this area. One of the compounds that's interesting is potentially the top compound here, where obviously we haven't got methoxys or other substituents on here, but we still get a kind of basal potency that's not bad. We still maintain quite good selectivity on the top compound. And then some of the other intermediate compounds have not worked as intended. Okay.
That 2022 might be an interesting one to put in the X-ray
yeah.
as well.
I'm sure we're going to talk about this a little bit later on, but can you see from the, you have a sense right now what's driving the selectivity between the two? Is there anything obvious that's coming up?
Yeah, there's some ideas, but nothing concrete. We're hoping that one of the structures, especially for the one with the methyl group on the inside position, once we get the structure of that, that may shed some light. But as yet, I don't think we have enough to say.
Okay.
Still very much in that learning phase.
No, I realized, I expected that I was going to be told I was reading the last chapter first. So.
Yeah, no.
So Liz here, pictured on the top right where you can't see her, but she's just been setting up some cell viability assays here in Oxford. So she's been using two methods, a cell titer glow assay in blue, and then also a PI assay, a propidium iodide. Is that right? Yep. Which is a sort of permeates dead cells, and then you can have a quantification of the signal there. So the sort of early data with reference compounds that are published agrees quite well with the literature, which is kind of just setting up the assay. So we've
Alex, those are around 2 micromolars?
Possibly 1 micromolar.
1, 2, yeah. Okay.
Around that.
Yeah.
So 1 to 2 micromolar, depending on the replicate. Yeah.
Okay.
I've not put in the IC50s for this, just because we're just developing the assay, and give us a week or two. So this is just taking just a couple of example compounds from the M4K series. Again, we're probably seeing what we ballpark expect in this 1 to 2 micromolar range. Some of them are a bit weaker, but I think they agree with what's being reported by other groups. Okay, so that's a sort of work in progress. I think I'm at the end there. Be happy to take any questions or to hear later on new priorities for cell assays or new priorities for crystallography.
Yeah, absolutely. Yeah, like I said, I think we'll need to touch base about some of the priorities for the crystallography. Some of the SAR is changing as we get new data, so I think it's always good to have these meetings. Okay.
Can I ask a question about the nanobrett?
Well,
yeah, so obviously Promega is a good friend of the SGC, and we've been collaborating with them often to develop new nanobrett assays. And what are you comfortable in my telling them about the performance of the assay? Because if their CEO is very passionate about open drugs and open science, and so it's a potential collaborative partner for M4K. And so obviously feel-good stories with their technology will help me. And so are you guys comfortable saying that this nanobread is a assay that we can use for in-cell target engagement, and we can generate SAR with it?
Absolutely. We've been developing this work with Matt Ropers at Promega quite closely. We've been exchanging emails, exchanging data. We're very, very happy with the assay. We're chatting with them about extending it to off targets. So out 1, out 3, out 4, out 5 would be nice. So that's a work in progress, and definitely we'll follow up on that.
Okay, thank you.
Great. All right, I think David, you are up. You grab your mouse there.
Okay. Let me just get back to the slide here. Okay, so this is going to be an update with regard to all the chemical matter that we've made to date, barring a few compounds which we have yet to send to RBC. So this first slide essentially summarizes, I think, the key SAR elements in the series that we've been looking at, which is this pyridine. So this is the initial compound which we've been taking forward, and this is the basis of our series. One of the compounds that we made here quite recently, actually, was M4K 1071, and this is a compound which Alex had made, I think, and published in the JMed Chem paper in 2014, but didn't disclose any ALK2 and ALK5 data, biochemical inhibition data on it. We did that at RBC, and what's interesting, actually, we find that the elimination of this central methoxy, it preserves the activity, so it's essentially equipotent to the initial compound, but the selectivity for ALK5 is quite dramatic. And this is not something we necessarily expected based on our knowledge of the crystal structure or whatever. So this is kind of a new result. This is an N of 1, so we're, let's wait to see, what more examples look like. But this is an interesting result in its own right, something we can potentially use going forward because it definitely simplifies the structure for us.
It's interesting that the nanobread IC50 is affected, even though the intrinsic potency isn't.
Yeah, absolutely. This compound was one of the ones which early on in the reports from SGC Oxford was sub 30 nanomolar, and at RBC, unfortunately, it's testing considerably higher. And so this is one of the things which is good to know for us, because we've essentially deprioritized the series where we have this methyl at the 2 position. So that was a good piece of data to know. We have deprioritized this. We're not going to be looking at this probably ever again, unless there's some new data comes to light which changes our mind, but we definitely won't be taking this forward in the near term. One of the compounds that we made early on here at OICR was to put this methyl group on the inside in the 4 position. And what's interesting here is that, again, we're equipotent essentially to the parent, but we're seeing a considerably larger window open up versus ALK5. So this is a really nice result in the sense that we have a methyl group which is going to give us freedom to operate. It's very kind of a proprietary methyl group, you could say, right? And it's something I think unique to kinase structures, kinase inhibitor structures, right? I don't think I've seen this kind of element used before. Why this group has this sort of effect on the selectivity, we have yet to determine, but a crystal structure should hopefully help us answer that question.
So just to follow up, and I did show this to our legal.
Yeah, I was going to ask for freedom to operate.
And they confirmed by looking at least at the Harvard patents that we do have freedom to operate with this methyl. In all of their patents, that position is only hydrogen.
Interesting. Is that also the same for 1071?
No.
Did you ask about that? About what freedom to operate?
1071 is covered.
It is covered. Okay.
And significantly here also this inside methyl, it preserves the activity in cells. So this is good. This is a good kind of feature that we can definitely work around. For M4K 1212, the story is a little bit more interesting. This data that you see here was obtained with the SGC Oxford legacy compound, which we had profiled at RBC. And the results look quite good, and we had started to initiate a number of plans around this motif, ortho, in the ortho position of this arrow ring. And in fact, we remade this compound here at OICR, and we sent it to RBC for screening as a batch 2, and unfortunately that batch is inactive entirely. It shows not even the slightest hint of activity. So one thing that we have to establish is why that's the case. Although Jeff will show some results a little bit later on in this talk, which may negate or need to fully explore this, because I think the PK results that we've obtained with this compound aren't ideal. So this may not be an overarching problem, because we may end up dismissing this subseries in general. So,
Alex, I noticed that you're still trying to crystallize the original batch of 1212. So that will be very interesting if we can get that structure. Yeah, and I think we also we'd like to see if you guys have any more of the original batch of 1212, so we can compare it with our material that we generated, just to make sure that they're both the same material.
How much did you send us?
I think it's really important that you identify, that you check the purity and the structure of both batches.
Right.
Yeah.
Right. Yeah. No, that's exactly right too. I think we do want to get to the bottom of this, because I mean it is quite potent. So or at least the original batch is quite potent in the enzymatic assays as well as in the cell-based assays, and so it would be good to figure out what exactly is going on here.
there's another piece of data later on in the presentation which I'll show you, which is suggesting that maybe in fact these compounds aren't as potent as we think. Yeah, I'll come back to that in a moment. So just one note about this information. I mean, summarized here, we're basically 80 compounds into this, and this represents the kind of the directions that we're following. And I think it's important to note also that when we've screened a few compounds against the mutants, but when we're seeing that activity on ALK2 pretty much guarantees good activity on the 3 mutants, and also activity on ALK1. So ALK1 and ALK2, they travel together, essentially equipotent. So we haven't yet seen a compound which shows marked selectivity versus ALK1. There's perhaps some little hints depending on how you want to read it, but I wouldn't read too much into that.
And we're not super concerned about that.
And we're not super concerned. No. Okay, so here's a little bit of discussion about this 4-position methyl group. So you can imagine that if we're seeing this kind of selectivity window, 150-fold and up, and we're seeing decent potency, what happens if we return that 2-position amino group, which we know gives us really good potency, but erodes selectivity in the parent? If we do put this methyl group in, can we bring some of that selectivity back? So this is one of the ideas we're going to be trying out here. Of course, you can imagine pairing this selective des-methoxy with the inside methyl, so maybe they'll reinforce each other, so you can have kind of additive selectivity. You can imagine bringing both of the elements together if in fact this does not erode the selectivity. And we've also been trying to explore different groups on this 4-position. Here's one example of a compound that we've made. We've also made the O-methoxy. We're going to put a fluorine there, chlorine, etc. So we're going to see what different bulk and different electronics at that position, what effect they exert on the selectivity.
And I think you're hoping that it'll improve the binding to the hinge binder.
Right. We're hoping that by pumping some electron density into this ring, we're going to have this lone pair here donating a better donator. This bottom series here is preserving this core methyl with this trimethoxy motif, but changing this solvent exposed portion of the structure. And you can see it's kind of it's a mixed bag. We have some with a little bit different geometries here. Not as good activity on ALK2. Here we're having a deletion of the nitrogen, so from a piperazine to a piperidine. Very potent compound. We're into the single-digit nanomolar now. The selectivity is very good, essentially paralleling the original, and also the nanobreadth is looking quite good. We have this compound right here, also single-digit nanomolar, decent selectivity, very nice nanobreadth. And this is an interesting example here in the sense that we've taken the parent and we've added an isopropyl group to the tail end here, and we really see some very, very nice selectivity over ALK5. So we're upwards of 300-fold now. And again, respectable in the nanobreadth, so we're under 100 nanomolar there. And again, single-digit nanomolar. So this is the first set of compounds where we're sub 10 nanomolar, and it seems to be consistently the case. So on the next slide is just a quick summary of what I showed you on the last one in terms of the series that we're seeing. So here's a nice example of the progression from, you have nitrogen deletion and the isopropylation, and you're having this progression in the selectivity while maintaining nanobread activity. Here's a legacy compound from SGC Oxford, which was very nice on the nanobread, decent selectivity. And here, although not directly comparable, because this one has an extra methyl group here, you can see that this, again, the inside methyl here is exerting its influence by opening up the selectivity window. So there seems to be at least a decently consistent pattern with that inside methyl.
Can I ask a question, please?
Yeah.
Just strategically, when do you chemists start to think about blood-brain barrier as, is it now or later?
I mean, do you want to?
Yeah, I was going to say, I think we're waiting until we see a little bit more biochemical potency. I think that, it would be nice to get sub 5 nanomolar, I think.
Okay.
Because we know that's possible in the kinase space, right? So it's,
yeah, you'll see as well that we're also bringing in the MDCK, MDR1.
Okay.
So
it's
we'll be
sorted out,
profiling
right?
all of the
It's
analogs that we generate in that, or most of the interesting ones anyway, in that assay. We'll bring it up later, but almost got it up.
So the other interesting factor is when you're adding alkyl group on the nitrogen, you are having better selectivity potency. It's good news for BBB.
yeah, absolutely.
Yeah.
When you have group here, it helps BBB penetration.
Okay.
It's very working in our favor.
Yeah, and an analog like that one with the amino as well, with the position on the purity, might bring back some of the potency and keep the blood-brain barrier and decent PK, right? So, again, every molecule we make is a step in the right direction.
In terms of potency, you're talking about biochemical potency.
Biochemical.
Is the nanobread actually more important? I mean, if you're looking at a lower.
Certainly, but we're finding that they generally track, right? So to be sub 100 nanomolar in the nanobread, it seems that we need to be in this 15 nanomolar and under in biochemical. So you're right, absolutely. Drives the whole deal.
Because 46 nanomolar looks pretty potent. To Alan's question,
Yeah.
44 nanomolar.
Yeah. Yeah, no, we're already are almost there. It's more of selectivity that, the full profile.
Right.
Yeah.
But we're getting
And freedom up.
Oh, sorry, just a moment. So we're getting signs of how to differentiate, which is really interesting.
Yeah.
So here's one of the things that Alex is working on for us, which is the crystallization of this guy with the inside methyl. So this inside methyl would live here, and we want to see if it exerts a torsional effect on this arrow ring and this arrow ring, and that may be what's responsible for some of the selectivity, or at least that's the operating hypothesis. And see when we do a structural overlay what exactly is happening there, and hopefully many other structures to come. So here's a series which is a little bit in question right now, given that this batch 2 from us was inactive. I think what's kind of telling here is that we've made this compound, except we put this methyl group up top. And what you'll notice here is that we initially had 9 nanomolar against ALK2, and now we've essentially lost selectivity. In the example, if I go back to this one right here, you'll notice that the methyl group up top in general, as I say, without the dense methyl was 30 nanomolar. You put the methyl in, you go to 115. There's a loss in activity, but it's not complete, right? Whereas if you go to this example, we put the methyl in and the loss of activity is complete. So this almost argues that this series was not active to begin. At least that's the thought, right? But we won't know until we stack the two, the initial legacy batch against our new batch. We'll try and solve that problem to see what the difference is.
If it is active, I mean, why not drop it? And because it's going to be a rabbit hole, maybe, doing the chemical analysis, showing that one's inactive, then you're going to go, we should check it again. And what's the harm in just saying, we're doing okay, why pursue this?
Yeah, I think mostly because the original molecule that we assigned to B1212 was so very potent.
Okay.
We would like to try to figure out what it is, if possible. If it's not this, and I mean as well, we'd like to compare with ours as well, right? So I think that there's enough incentive, and I mean we won't go too far down the rabbit hole.
Yeah.
But if we can, obviously it has the same mass. So it's
Hey, this is Peter. Were both batches made by the same row?
That I don't know. I don't think we have the chemistry route that was used at SGC.
We don't. Or at least we certainly didn't look at it.
Yeah. But, I mean, this is data that just came out last night, and we just figured out this dichotomy this morning. So, you know, I think we can take this offline a little bit and try to figure it out in the coming weeks. You know, this stuff, but it highlights the importance of kind of resynthesizing some of the important molecules to make sure that, you know, we get similar results. So
we have a couple of compounds in queue still yet to be screened at RBC, which may speak to this issue also. I mean, we have the morphine variant of this, which we don't anticipate the potency should be that different because it hasn't been in the past from prepared. So we'll see what happens here. And, you know, we had started working around the series, doing some docking and looking at some functionalization of this ring, which fits into a little pocket near the hinge. So let's see what these guys look like, right? That may tell us, right?
If they're all inactive, then
Clearly, yeah.
And again, Alex is trying to crystallize the original batch of 1212, so we may get some hits there.
What the structure is.
With the structure, yeah, exactly, right?
Okay, so as far as, you know, one of the focuses now that we see that we can eliminate that one of those methoxy groups on the trimethoxyphenyl, that will be certainly a direction we'll be following. But one of the other directions we were following right from the start was, can we replace that entire trimethoxyphenyl motif? Because there were some considerations potentially down the line in terms of metabolic liability, et cetera. So we had started kind of really exploring quite widely with a considerable degree of freedom around this pocket side group, and not much has come of this. I think this is arguably the most interesting compound to arise from that exploratory set, which is this para-amide. Very nice selectivity over ALK5 and decent potency at 31 nanomolar. Don't have the nanobread data because actually we didn't make enough of this compound to send, but this is something we can do a remake and send to see what this looks like. And of course, you can imagine that, you know, adding the amino group back might bring the potency down, and we can explore the O-methoxy anchor. So there's room to operate around this kind of motif with the amide to see if anything can be gotten there. Some of the analogs that did go in with this first one, you can see this when you put it in this meta position, it's inactive. And when you move this fluorine from this para to this meta position with respect to the amide, you can see again a little hint of activity, but nothing too spectacular. Another somewhat interesting compound was using this methoxy as the anchor and this para nitrile, not too bad at 80 nanomolar, but again, probably not worth pursuing at this point because we do have better things, and we can play around a little bit and see if we find some improvements there. So this is a really nice, Dave McLeod was working on this, and this I think evolved from the RIPK2 data that was presented at the meeting last month, where it was shown that the RIPK2 compounds did in fact show activity on ALK2 also. And one of the features that was noted in that work was that when you preserve this R1 methoxy as a critical anchor, delete this R2, which we turn into a hydrogen, and then explore around this R3, it's discovered that, especially in the case of the RIPK2 compounds, you could put groups such as sulfonamide in there, some fairly large, bulky moieties, and you could actually have activity on ALK2 still, which was unwanted in the RIPK2 case, but for us, it's something we can take a look at. So Dave McLeod did a nice study where he took this oxygen and he substituted on a variety of R groups, which appear to dock quite well in the model that we have. But unfortunately, and interestingly enough, when you go from just the single methoxy here, which we've now demonstrated is quite potent, you add a couple more carbons, and we start to get the activity up above 100 nanomolar. And when I say above 100 nanomolar, we're ranging basically from 100 through to like 500, 600 nanomolar, based on whatever the bulk of this group is, but not good enough for our purposes, right? So this was a little bit of a surprising result, given that we know from previous work that, in fact, considerable bulk is tolerated at this position. So we're going to go back and we're going to take a look at some of that RIPK2 work and make some of those compounds and actually screen them in our hands, see what's up there.
You guys have probably said this already, but can you replace the blue OR with the halogens, which is chlorine?
That's also in the to-do list. Yeah.
Okay.
Yeah, that's exactly next on the.
And I know people often don't like acetylene, but it is in Tarceva.
That's right.
In a similar electron-rich ring.
Absolutely.
That might be a good item here as well.
Yeah, for sure.
It really seems like having that one methoxy is key. Yeah.
Mm-hmm.
And
then you can play around.
you can play around a little bit with the other substituents.
Couple of compounds still in the queue here. You can see changing one of the methoxys to a slightly larger CF3, so that'll be interesting too. And also you have a little bit of a different electron clouder on that, so I'll see what effect that has. And putting a fluorine in the middle between the two OMEs, which should hopefully parallel this compound. Again, more ends, so we know where we fit. And just one last slide to finish up here. I just want to make the isomer of the core, so moving the nitrogen and imperial, and this is dead, so that's nice. It basically locks us into the one imperial core that we're used to. And then there's some interesting, a little bit more exploratory compounds, bringing back that top amino, which we know confers activity, but can we lock it down inside a ring and see if a scaffold like this can maintain that activity, maybe find some selectivity and potentially another series to open up. So that's about it for now, and yeah, we have compounds in queue, so hopefully better things to come. That's it. So,
Great.
any questions? Yeah.
Owen and I can't help much on the chemistry, but if you're looking for help on considerable bulk, we're in there.
Dragging me down with you. So that's fantastic. I mean, sure, before. That methyl is a great example.
Yeah,
Yeah.
it seems to be magic.
Magic metal. Well, another magic metal. So is this the? Oh, go ahead.
Yeah. Okay, so we just wanted to kind of wrap things up by highlighting where we were at the last meeting and how we progressed those compounds and you know, really how we'll move things forward throughout the project. So, you know, we had presented this SAR with 1046, 1055, and 1062, which is the amino, the methyl, and the hydrogen. And, you know, when we looked at all this data, we really felt that, you know, 1062 gave the best balance between potency, selectivity, and properties. And moving to the hydrogen was a good idea. And then this 2009 was also a relatively new result that we had tested against ALK1 and ALK2, and it's potent, and it gives some good activity in the nanobret. And so this 4-methylpyridyl is a really great finding, as we've highlighted, and it gives us freedom to operate right off the bat with a very simple modification. It doesn't increase the molecular weight relative to 1055 at all. So, you know, again, we're following up on this, and things look quite promising. And then we were originally very excited by 1212 because of this very promising overall profile. Again, though, when we remade it, it seems like this you know, there's more work to go into understanding what exactly is going on there. And just to remind everyone about how we profile compounds and what the plan is. So we have synthesis going on at both OICR and Charles River, and generally we come up with ideas, we model them, we dock them into various tools that, you know, Charles River has their own set of tools, and Gen has provided us with a set of tools here at OICR to be able to dock the molecules, and then we prioritized ideas in collaboration with Charles River. And it's interesting that, you know, when we all put ideas down on paper, similar ideas came out from both organizations. So I think that's a great sign, and we're all moving in the right direction. Then we go into the biochemical assays at RBC against ALK1, ALK2, and ALK5. We did spot check against the ALK2 mutants, the DIPG relevant mutants at RBC, and as David said, they track very well with the ALK2 potency. So, you know, I think we need to spot check every once in a while when we do something outside the box here, but you know, generally tracking this potency, you know, should do very well. Then SGC Oxford, as Alex showed, he's generating some X-ray structures. They're producing the protein. They're generating all the cell-based data in the very promising nanobret. I think the ALK5 is still being done in luciferase, but I think that's being worked on. So that's very interesting. And then the compounds don't actually come back here. We already have them, but they move forward in OICRs, in vitro ADME profiling, where we're looking at liver microsome, mouse liver microsome, and human liver microsomes. The MDCK NDR1 is almost ready to be brought online. I don't know if you want to make any comments there.
So yeah, we have the cells now, and we have the plated. They are plated. We're just doing the control compounds to see if it's working. Soon we can put in the compounds.
Yeah. So that will help tremendously in terms of looking at the potential for blood-brain barrier potential. So that's going to be brought online soon. Alex has just started presenting some data on the proliferation in the DIPG patient cell lines, or in the various cell lines. So that's going to be important to drive the project as well. The molecules that we've looked at so far are all around the 1 to 2 micromolar range. We'd really like to see that come down substantially. We also want to make sure that in cells we're engaging the target as we think we should by looking at the phospho-SMAD, the ID1 levels. RBC has agreed also to test a couple of compounds per month against a larger 5-kinase panel screen. We have not capitalized on that yet, but I think we're just about ready to send a couple of more promising compounds to see, test against a slightly broader set of kinases. And then, once we get some data here, we'll move the compounds into in vivo experiments. And the first one that we do here at OICR is a rapid PK screen, essentially, where we dose the compounds orally at 25 mg/kg, and then we only look at a couple of different time points over 24 hours to be able to just look at oral exposure. We don't look at the IV arm right away. I think this is something that we can look at later. What we're really looking for are molecules that provide us with good oral exposure. And I'll show you some data that we've generated on a couple of these analogs already in the next slide. Moving on, I think there's still a lot that we need to work out and figure out as a team in terms of the in vivo PK, PK/PD, and MTD studies. Where are we going to do this, and how are we going to do it? Scale up the compounds to be able to do that. So, but we do have time. It's early days. I think we're still several months away from this. So I don't think it's worth discussing at this point. But I think we should definitely start thinking about this in terms of where we're going to have all of this work done. So
actually, if you can just go back for one quick sec, for the prioritizing some of the in vitro ADME, but also this kind of goes back to what Al was saying as well. Is it worthwhile taking a look at any of these series now in vivo to take a look at any of the plasma brain penetration? Because this dovetails with where we're going to redo our budget very shortly. So the kind of reason, should we plunk in for 2 months down the line that we're close to a couple of these series that we might want to see if there is some indication of what the right brain penetration. Theoretically we know, but in practice it's a different thing.
Yeah, no, I think in 2 months, 2 to 3 months time we'll be ready. Yeah, at least to push in a couple of exploratory compounds. We may even have better than exploratory compounds at that point.
It's interesting too.
But I would wait 2 to 3 months so that we get we want n equals 2 on everything, and you'll see a couple of things on the summary slide.
But we're getting pretty close to 2 or 3 for some of these from what I'm looking at.
Yeah.
Seems like it's not a bad idea because also, MDCK we hope will be predicted, but we don't.
We'll need to verify that too.
Yeah.
Right. So once we get that online, then
So I think it's a good idea to do few compounds, but again, we don't have to rush it. But once we have the compound that we are comfortable with the compound, we should put them in vivo, do the in vitro assay, which is the MDCK, and validate is it translating.
Correlation.
Coordination is there as well.
So yeah, I agree there is a slightly different time frame from my perspective, because I have to submit a revised budget, and this was not gonna happen for maybe another 6 months. And so if people believe that this is something I should move up, I'm gonna just rewrite the budget and say that we're gonna start doing this in 3 months. I guess that's my action item. And that seems to be a lot of nodding going around the room. So
Yeah,
in our action, I think we're on cue that we should start looking at that.
Yeah, no, I think, like I say, I would give the SAR a little bit more time to ferment.
Yeah.
But, I think we're definitely moving in the right direction. I think we will have 2 to 3, maybe even 4 analogs that we can move in there. It would be good to have a decent number of compounds to try to get that comfort with the MDCK MDR1 results. It does correlate with what we're seeing in vivo with this series of molecules.
But just to make sure, so in the budget, we'll say for the end of next quarter, we may be doing the in vivo, not 2 to 3 quarters down the road. That's all I want to make sure that we have the money lined up on that side.
Yeah, I would say probably the end of next quarter.
Perfect. And that's what
We would select the compounds
And in vivo's done in a mouse?
Yeah.
So people ever do the in vivo studies in the xenograft at the same time?
More expensive. Usually what is,
Why expensive?
well, because you have to wait longer. You have to make sure the xenograft is established. These two,
and
But he's saying, yeah, if you're gonna do the in vivo efficacy anyway, you just check the brain barrier, right?
Yeah. And then this gets back to my planning point now. There are no commercial vendors for this particular disease xenograft. It has to be done in an academic lab, presumably, I'm guessing. So you better, they don't have these things like growing all the time. The mouse may not be, colony might not be big enough. I think it's wise to, I don't know what the heads up period you have to give to an academic that's running a xenograft.
No, it's a good point.
Start now.
Yeah.
If you guys are thinking 2 months you might be ready, then you go 2 months, all my graduate students away, the mice are sleeping.
Yeah, the blood-brain barrier penetration experiment is relatively simple compared to a full efficacy. The compound requirements are much less. It's a single dose. You look at plasma versus brain, and we would want to make sure that we get into the brain.
I'm just thinking, do it in sequence or do it in parallel, and if we do the proper thing in sequence and then do the thing in another 100 kids died, right?
So let's maybe we said that aside. I'm just kind of confused. Like from an antibiotics history, you start doing ADME, and before you really know the ADME, and you start doing efficacy, you don't really know what drove the efficacy. Sometimes you kind of have in between. So nail down a bit of the PK dynamics before.
But along those lines, it would be great if we could start figuring out where we're going to have those antibodies done.
Yeah, that I agree.
And really start, because the potential I see is that if we run blood-brain barrier penetration in 3 to 4 months, we'll be able to select 1 or 2 analogs from that study that warrant moving forward.
And for a social engagement perspective, wherever you guys think this efficacy xenograft stuff, we should get that professor and that postdoc engaged now so they feel they were part of that PKPD. I'm just trying to get them excited about it rather than come at them, Professors don't think like that. We need to be coddled. So I would suggest
that they're on the line, right?
Yeah.
This is a live mic, right?
But to be fair, right? It's a science project. We do want to.
We want to move it quick. So maybe Alex, we can talk offline another time about who we might think about having run these models.
He did forward some information.
Right. Yeah.
So we have to work on that.
Yeah. Okay. Great. Discussion. Okay, so the oral PK study. So I told you we run this rapid PK study in mice in-house where we look at only a couple of time points. We dose at 25 mg/kg, and we look at 30 minutes, 1 hour, 2 hours, 6 hours, and 24 hours to look at the plasma concentrations. And here we're comparing, or just showing the microsomal stability results that we have in mouse. So M4K 1212, this was the batch that we generated in-house. that we dosed, the one that was inactive that we just found out. And this analog doesn't show very good exposure. You know, very, very low. It's, 20 to 30 nanomolar. Kind of goes up and just kind of stays there. Whereas the exciting analog, we think that the one with the 4-methylpyridine shows, pretty good exposure for a dose of 25 mg per kg. We're getting a Cmax up around, 1.7 micromolar, and the molecule sticks around out to 24 hours. So, QD or BID dosing is certainly a possibility with analogs like this. So we think this is an exciting result. We still want to profile.
Can I ask a question? Sorry.
Yeah.
Sorry. Are these total concentrations, or do you do any protein binding at all? Plasma protein binding. So these total concentrations or free concentrations?
Total concentration. We precipitate everything out. All the plasma protein is precipitated out, and the small molecule is extracted out. We do a precipitation crash. So yeah, it's the total concentration.
Right. We haven't looked at protein binding yet.
I have a question.
Yeah.
Was the PK done on that 2009 on a free base or on a salt?
So they're all free bases.
Free bases.
Yeah.
It looks like there is some issues with dissolution probably.
Yeah, it seems to go in and just slowly absorb, right? Yeah.
6 hours.
Yeah. Yeah. But I think it's as a first step in this series. I think it's pretty good.
Great.
Yeah.
Have you thought about doing a similar sort of thing, but either IV or IP dosing? Just perhaps looking at the first 3 time points to look at your brain and plasma concentrations, just to see whether they're equal.
So, I mean, that's what we do for BBB penetration. We just take but once they usually complicates the BBB, we like to do perfuse brain, because as you know, brain has 10 to 20%, 4 to 5% blood in there. So you need to do perfuse at any, so those are not a simple one, just to take, poke the mind and get the blood. It is what we are doing right now. But that's the BBB penetration study we do. As Jeff was mentioning, we are going to do that. And because we are trying to develop the oral drugs, this is why this is like a screening, and we are going through we can go through a lot of compounds if we are doing in this way, and figuring out if we have the oral, candidate. And then we can always, IP is going to usually give you higher exposure.
Right. Are you surprised by how long it took to get to the highest level? 6 hours?
It does seem quite a long Tmax.
So there can be 2 things. One, as Diana said, maybe there is a big resolution problem. It's a pre-base we use, so maybe it was just staying there. And the other thing is maybe there is not a huge difference between 2 hour and 6 hours. See, once it goes in there, I mean, you're seeing the difference here, but once it goes in there, it stays there for some time. It doesn't clear as quickly as we.
Right. I guess what you say is between 2 and 6 hours. There could have been a Cmax in between as well, right?
It just seems like
it's on its way down.
These were mice, right?
Yeah.
I mean, the cycle isn't the cycle for a drug or passenger mouse. They're like just through the intestinal tract.
Well, in 2 hours, 2 to 3 hours, it goes through all the way from.
Yeah, so that's why it still seems to be growing at 6 hours.
We are doing it at 6 hours, but as just mentioned,
the peak was really something.
Yeah, somewhere in the middle,
like 3 hours or 2.5.
It could be.
Yeah. Okay.
It could also be, as Ken was mentioning, a solubility effect when you dose the molecule. We put it in a solubilizing formulation containing a small amount of DMSO to make sure that it's completely soluble when we dose it. And then you dose it, but it could crash in the stomach.
Yeah.
And then just slowly
I think Alex mentioned in one of his nanobreath summaries that a couple of the compounds, none of the ones that I showed, but a couple of the compounds form cloudy solutions. So I think we were discussing, Dave and I, that maybe some of these are kind of on the cusp in terms of their solubility. So that may be something we have to kind of keep an eye on.
Yeah.
So that may be...
I think someone mentioned that forming a salt would certainly help with your solubility.
Yep.
Yeah.
We can do that.
Well, we got a lot to learn about the whole series. So.
Yeah, I mean, these are snapshots that we try to get to prioritize within the series and to try to learn as much as fast as possible.
I mean, it's still great results, you know, we're getting some interesting data.
Yeah.
Trying to understand.
That's exactly. So this is
That's the takeaway. It's exciting data.
this is just a screening tool for us. This is not the usual result.
Right. This is our rapid in vivo series, right? So we try to get as much information as fast as possible, as many compounds as possible. So we'll move, several compounds through this.
And the stability is looking quite good. I've always mentioned that the barrier is quite high because we're doing it in an hour, right? So the stability usually is done for half an hour. So the compounds look quite stable, I think, generally speaking.
Phase 1, yeah.
Kind of another stake in the heart of 12-12 though, isn't it?
Yeah, definitely is. So I kind of put 12-12 in red now because I just got this data this morning, so the slides were pretty much all ready. I think it's important that we do talk about, what are the next steps for some of these compounds, and, certainly 12-12, 2009, the methyl compound, and then the two reference compounds as well, with the methyl and the hydrogen. We want to move those into these rapid PK screens so that we can benchmark against those. How far, because we know that both of those compounds showed really good PK profiles in a mouse. And we know that the methyl compound, I'll just come back to it, everyone knows. 1055 showed good brain levels after oral dosing. So, I think we want to know how we're comparing against these two molecules so that we can benchmark against it with our in vivo work. So as I mentioned, 12-12 and 2009 have been dosed, and then 1062 and 1055 for benchmarking will be dosed in the next month or so. X-ray for these compounds, certainly up 2 and up 5. This is, as Alex mentioned, is ongoing. I think, Alex, some of the other compounds that we had originally selected, we can talk about potentially deprioritizing, but I do know that, many of these are going in already, and some of them we already have X-ray structures for, so certainly 2009 we think is the highest priority analog for us in terms of X-ray. During the chemistry meeting that we had with Charles River, we talked about potentially profiling some of the analogs in SIP and potentially HERB. This is something that came up here after the meeting, and this is something that I think Stu is looking at Charles River, whether or not we can try to have some of our analogs profiled there. I think we can follow up on that at the next meeting. But that would be nice to be able to start gathering some of this information on some of these series of molecules or sub-series of molecules to make sure that we're not going to identify something later. I think as well, the data we're getting from RBC is fantastic, and I think we're getting really good alignment with that data and what we're seeing coming out of Alex's lab in terms of the cell-based data. So that's great, but I think it is important that we get N equals 2 for the most important compounds at RBC and in Alex's lab as well. With on different days and separate experiments, just to make sure that we're following the SAR and that we're not misled. And then, profiling in patient-derived cell lines in terms of proliferation phospho-SMAD. Alex started showing some data today. I think, that looks extremely promising, and look forward to seeing a lot more data there as well. Alex introduced us to Jerome Fortin, who is a researcher at UHN, and he's also got some primary DIPG cell lines that he's willing to test our compounds. So we're trying to put an MTA in place to be able to enable that. And again, I know that Nana as well has access to some of the DIPG cell lines, and is running some of those assays in her lab. So it'll be great to run our compounds against as many of these cell lines as we can get our hands on. And then, certainly profiling some of these more novel analogs against a larger kinase panel. This is something that should happen relatively soon with some of these more advanced compounds, and we'll pull the trigger on that in the not too distant future. And then, as we were talking about, a mouse full PK profile, looking at brain exposure, and then an efficacy study. Also need an MTD study in there at some point as well. But I think we're moving in the right direction. We're moving toward that, and I think from the discussion we've had today, we need to start thinking about this a lot more carefully in terms of where and how we're going to have this done. We need to continue to profile the most promising analogs. I think David gave a great presentation about the SAR that has just come out. We need to digest that a little bit more and continue along profiling some of those more promising compounds. MDCK, NBR1 is almost up and running. We should have that very, very soon. Big thing for us is finalizing the database and establishing data sharing protocols. That's still something that is, that needs to happen, and we are working on it. And I'm hoping by the next month that we do have this, and we can present something showing exactly how we can share data effectively across all of the sites. Right now, we're doing it piecemeal, and again, the number of compounds is low enough that it's not overly burdensome, but it's getting there. So this does have to be resolved relatively soon. And then certainly I think we need to continue the SAR. I think we're generating some exciting results. I think these are exciting initial results, and I think with a couple more iterations on the SAR, we're going to have even better analogs that we can move forward into some of these assays. With respect to the 4-methyl pyridines, I think that the freedom to operate is certainly a very positive aspect, I think, about that molecule. It's potent, it gives good PK, and we have freedom to operate. So, we're hitting many of the benchmarks that we need with them. So. And then, some of the trimethoxyphenyl replacements, that seems to be a more challenging part of the molecule to address, but we have, I think, some exciting results there as well. I think this fluoroamide is interesting. I don't know about blood-brain penetration, but certainly as an initial example, that molecule showed absolutely no inhibition of ALK5 at all. It was completely, we showed greater than 2,000, but there was no curve at all. There was no inhibition of ALK5 at all. So I think that's very positive, and using SAR like that in additional analogs, I think we should be able to move things forward pretty quickly. So with that, I think we're pretty much on time and on target. And if there's any questions, any additional discussions?
One quick one. Does that freedom to operate allow for any substitution there at that position?
Yeah, I mean, in the Harvard patents, it was only exemplified and covered as a hydrogen.
So maybe a couple of other substitutions at some point could be explored as well.
Yeah,
we're on, Marty. We have the O-methoxy's done. The chlorine is in plan. The NH2 is done, right? So, and if the NH2 is viable, you can imagine analogs if you want, right?
Yeah, potentially cyclizing between there and one of the other rings on either side could be interesting as well, which would give us freedom to operate and maybe really tune the conformation to what we need, right? Depending on the ring size. So I think that there's a lot of opportunity there.
So you're thinking about chlorine too, right? Chlorine there? Are you
chlorine, yeah, that's in plan. All of the
chlorine.
Chlorine, sure, yeah, absolutely. We can run the whole.
It's about the same size as the mountain.
Yeah, exactly. So we can run the whole.
All right, great stuff, great progress.
Can I just give a short? So, this is just FYI, there's no actions for anyone on the call. There are genetic evidence that the PRC2 complex, the sort of EZH2 is involved in this tumor. There are no compelling data yet that show that an ALK2 plus an EZH2 compound will be synergistic. I think Alex is trying to get that going. But in anticipation of that, I'm starting to reach out to the companies that have these EH2 programs, none of whom are developing them for brain cancer, just to see and explore if there are ways that we could take some of their knowledge and apply it to this, or eventually even make a publicly available open EZH2 compound specifically, if the data continue to show that they might be synergistic. Okay? So Pfizer, Glaxo, Epizyme are the three companies that I know of that programs. So I'm going to give them each calls. If Pfizer's here next week, I might chat with them. Okay?
Okay, fantastic. Any other questions on line? EZH2. Okay.
No.
Okay, great. So thanks everyone. I think this was a great meeting again, and look forward to next month's meeting.